Now we talk about what several member protein that can help to transport specific molecule. First is ATP power pump and ion channel and the transporter. Okay, so first I would like to introduce how you read this graph. So this is triangle. This is the gray is the membrane, and this is triangle. Is mean there will be higher electrochemical gradient of those red molecules from this side to the other side. Okay, and if you see the gray triangle, it means the black molecule here have higher electrochemical gradient to the other side. Okay, there's a principle of this events. If the molecule transport from the higher just follow their electrochemical gradient, they don't need the energy. If they transport against their electrochemical gradient, they need the energy. Okay, so if this molecule transfer from the triangle of this side to the other side, they don't need the energy. But if they against this direction, they need the energy. Okay, there's no exception. Okay, so the ATP power pump, they use ATP hydrolysis as an energy to transport molecule against their electrochemical gradient. Okay, and the ion channel, they don't need energy. They just kind of change their conformation and open a tunnel for molecule to transport, follow their electrochemical gradient. They don't need energy. Okay, but but for the transporter, if those are protein or transporter only transport one kind of protein, we call it uniporter. Okay, but uniporter usually they just transport molecule follow their electrochemical gradient. They don't need energy to transport molecules. But the symporters, or we call co-transporter or antiporter, co-transporter, they transport multiple molecules at the same time. Okay, in this case, symporter, they transport two molecules in the same direction. They call it symporter. If they transport two molecules in the opposite direction, people call it antiporter. Okay, so symporter, what kind of energy source is? They will transport the red molecule, follow its electrochemical gradient, and use this as an energy source to transport black molecule to against their electrochemical gradient. Okay, I will let you know the detailed mechanism later, but you just need to know the definition. Okay, but the antiporter, they will use the energy. They will transport the red molecule, follow its electrochemical gradient, and use this as the energy to transport black molecule to against their electrochemical gradient. Okay, the energy, these two guys and these guys need energy, but they use a different energy source. These guys come from the ATP hydrolysis, and the co-transporter, the energy comes from the molecule they transport. For example, in this would be red molecules. Okay. Or basically, let's just, for example, the red molecules, I can briefly explain how they can use this as energy. Just this red molecule, they transfer from this side to the other side, they will change the conformation of this symporter or antiporter. And by changing this conformation, it needs energy. They will gradually transport the black molecule against the electrochemical gradient, something like that. Okay? Yeah. But this ATP power pump also need to change their protein conformation, but those are protein conformation changing driven by the ATP hydrolysis. Okay. So depends on whether they need the energy investment or not. If you need energy, we call the active transport. Okay. For example, this will be the ATP power pump. We need its active transport, and also these are two guys against the electrochemical gradient to transport molecule. They also call the active transport. But you need to kind of distinguish this part and then this part. So from some textbook, they call it secondary active transport. It's kind of boring, but yeah, just definition, secondary active transport. But for these two guys, they don't need energy, so those are not active transport. But they do provide some help for their transport. So we call that facilitated transport. It just provides the easy way for them to transport, follow their electrochemical gradient. Okay, like aquaporin. They don't drive the movement. The driven force is their electrochemical gradient, but they provide the tunnel or path or shortcut for them to across the plasma membrane. Okay, so uniporter can do this. So you can imagine if the electrochemical gradient of this red stuff opposite, the molecule will transport from this side to the other side. Okay, they just open the door, open the gate. They don't decide the orientation. It's also the case for the uniporter. Okay. Okay, so multiple channel, those member proteins, they actually work together because they share the same. Usually, if those members connect with each other, they share the same electrochemical gradient. Okay, yeah. So for example, this is a sodium-potassium pump, ATP power pump. They use ATP hydrolysis as an energy to transport a lot of sodium to the extracellular space and transport a lot of potassium to the cytosol. So after a while, they will generate the electrochemical gradient of the sodium and the potassium of the whole plasma membrane. And the other ion channel or those important can use these electrochemical gradients to deliver the potassium from this side to the other side. And also can transport, symporter can transport sodium from this side to this side. Follow is the electrochemical gradient generated from this ATP power pump. Okay, so they share the same system. Okay, they work together. So this is a table for the definition. So it's a simple diffusion, facilitated diffusion, a transport or the active transport and the cold transport or cold secondary active transport. So simple diffusion don't need any specific protein. For these three mechanisms need specific protein. Okay, and only these two need the energy. So it will against is a gradient. That is why it need the energy. Okay. And these two, this energy comes from the ATP hydrolysis, and this comes from the co-transported ion down is a gradient. Okay, that's it. It's really clear if you can get my point. Okay. Okay, so we finished the section 1 of the chapter 11. So the large and the hydrophilic molecule, they are difficult to diffuse across the membrane. This means the pure phospholipid membrane. You need to know the basic chemical property, okay? They are not permeable to the hydrophilic and the large molecule, basically, okay, and the charged molecules. And the several protein can help to transport specific molecule to the cell or outside the cell to generate specific environment to run those physiological reaction. So ATP power pump, ion channel, transporter, and then we will introduce them one by one in the following section. Any questions so far? No? Okay. So first, uniporter. To understand uniporter, we would like to kind of give you a comparison between the passive diffusion and the uniporter. So for the simple diffusion, the rate is slow. The uniporter transport is fast, okay, because it generates shortcut. It generates specific tunnel for them to kind of, they don't need to deal with the chemical property of the phospholipid, right? They provide the tunnel, they provide the shortcut, so it's faster than the simple diffusion in most of the case, okay? And the specificity, simple diffusion is non-specific. It all depends on the hydrophobicity and the size, no matter what kind of molecule it is, okay? But the symporter is specific. Uniporter is specific. It only transports certain molecule. For example, we will introduce one example, is a glucose uniporter. It's only transport glucose. It will not kind of transport molecule, depends on its size and depends on its hydrophobicity. It only transports certain molecule because they have a kind of a binding site of that molecules, and that is why it only receive or generate the shortcut specific for that molecules, okay? And the direction is reversible. What does this reversible means? It means that the transport can go this way or the opposite way. It totally depends on the electrochemical gradient of that molecules, okay? They don't decide the direction. The electrochemical gradient of those cargo determine their delivered direction, okay? So this is an example for the uniporter, which is a group 1. It stands for the glucose transporter, okay? So the glucose have a higher electrochemical gradient in this side compared to the other side. So this is the GLUT1. Basically, those are unipolar. They just keep a changing layer conformation between this conformation and this conformation. And they have a glucose, in this case, they have a glucose binding site. And those glucose binding sites will not continuously bind the glucose. They will keep, the glucose will keep shuttling between the pool, cytosol pool and that binding site. So they will keep binding and the associate bind and the associate, okay? So those GLUT1 will keep changing this conformation, and those glucose will also bind the associate. So if glucose have a more or higher electrochemical concentration here, there will be more glucose bind here. Since this GLUT1 will change their conformation and they will open to the other side, they will also release the glucose. After a while, they will transport this glucose. Depends on its electrochemical gradient. Okay, you get my point? Yeah, it's a really basic mechanism. It just keep this binding site will not continuously bind the glucose. It will have a weak affinity. Okay, so it will bind, release. That is why you bind and open to the other end, they release. Okay, if the glucose have a higher electrochemical concentration here, they can do the opposite reaction, deliver direction. Okay, and also we will kind of introduce the other kind of not only electrochemical gradient. When you talk about water, we need to deal with the osmotic pressure. 渗透压, okay? So what is osmosis? So osmosis all happen to the case which have, to the system which have a selectively permeable membrane, okay? That system, it needs a selectively permeable membrane, which is our plasma membrane, okay? In this U-shaped tube, there is a selectively permeable membrane, and those are green dots, they are glucose, okay? And the blue are the water molecules, okay? So this selective permeable membrane, they are only permeable to the water, and the glucose cannot go across, okay? So glucose are hydrophilic molecules. They are easily dissolved in the water. It means chemically, the water like to bind the sugar, okay? That is why the glucose can dissolve in the water. If the one chemical, they are hydrophobic, and you mix them with the water, after centrifuge, they will form a pellet. If they are hydrophilic, they will dissolve in the water. After centrifugation, you cannot see anything. They dissolve in the water, okay? It's also the case for the sugar. The water like to bind the sugar because sugar is a hydrophilic molecules. But you can see in this, there will be two pools separated by this selectively permeable membrane, right? As I mentioned before, sugar cannot go across. Water can freely go between these two pools. So water can go, but sugar cannot. And then you put more sugar here, less sugar here. What happened? So how to deal with this difference of the sugar solution? Just water, more water will come from this side to the other side because they need to bind more sugar, right? Because they are more sustained. So they will go, more water go there because they are more water sugar can bind, so they will go there. Because glucose cannot come here. That is the only way to deal with the water go there. So the water will go to bind more sugar here. That is why it will generate such a water level difference. That's the osmotic pressure. Okay, it's all about water. It's all about whether those are more substance water can bind between these two pools. Okay? Yeah, so that is why our membrane is also a selectively permeable barrier. Sometimes the cell will kind of encounter to the isotonic solution, which means 等张压力, which means the substance outside the cell and the inside the cell are comparable. So the water will not go out, and there's no net movement of the water go out or go in the cell. So the cell will keep the normal shape. But in the hypertonic solution, 高张溶液, okay, in Chinese, there will be more substance outside the cell. So substance cannot across the plasma membrane. The only way is the water go out. Okay, so the water will go out to bind more substance. Then you will lose water and the cell will become shrink. Okay, but if you put the cell to the hypertonic solution, 第一章容易, there are more substance inside the cell. They will absorb more water to balance such an osmotic pressure. Okay, then the cell will become swell and finally ruptures. Okay, so this really danger for a lot of microorganisms like yeast or some single cell organism. So they sometimes easily encounter to those hypertonic environments. So that is why they will generate cell wall to cover their membrane. Okay, they will keep the normal shape, and even they absorb the water, the cell wall is really sticky, so they will keep the reasonable size. Otherwise, they will kind of easily rupture. Of course, the cell wall will also provide some mechanical support. For example, someone step on them, they still can keep the certain resistant force, right? Okay, yeah. Okay, so when we talk about the hypertonic, isotonic, and the hypertonic, we call this a tonicity, okay, which is the ability of the surrounding solution to cause a cell to gain or lose water. Okay, but again, when we talk about the osmotic pressure, this is only happen to the water can really go across the membrane. But in the beginning, for the pure phospholipid bilayer, the water only slightly permeable to the pure phospholipid, right? So is it possible the water can really kind of easily go out and go into the cell? Yes, it can. It needs help for the protein I just introduced before, aquaporin. Okay, people found this and found the mechanism who got the Nobel Prize. Okay, so this is the monomer, the single protein unit of the aquaporin. They basically are a lot of alpha helix here to help them to embed to the plasma membrane. But inside those alpha helix, there is a tunnel here which is highly hydrophilic. And the diameter size of this pool is about 0.28 nanometer, which is only slightly larger than the water molecules. So the water can easily go across this hydrophilic tunnel. Okay, but you may ask, how about the smaller hydrophilic ion like a proton? No, proton cannot do this because they put some charge amino acid on the wall, so the charge like proton will stick on the wall, so they cannot really completely go to the other side. Only water with such a minor charge molecule, hydrophilic charge, can go through this tunnel, which is a really elegant design. Okay, so from the top view, this is only one monomer of the aquaporin, and they usually will work with 4 aquaporin together. We call it tetramer. So from the top view, this is the tunnel. Okay, so each monomer have one tunnel. So the whole complex will have 4 tunnel for the water to go in and go out. Okay, and which osmotic pressure decide the water will come from this to here or here to the other side. Okay, it's not electrochemical gradient, it's osmotic pressure. This is a cartoon, so you will see this size is a beautiful, perfect for the water molecule. They can gradually move from this side to the other side, depends on osmotic pressure. Okay, but the surface, again, the surface of these aquaporin are hydrophobic. That is why they can embed to the membrane. Okay, so the unipolar compared to the simple diffusion is faster, is reversible, and is specific, only transport specific molecules. And we use 2 examples for the unipolar. First is the GLUT1, which will change their conformation to transport glucose down, follow its electrochemical gradient. And the aquaporin provide a tunnel or channel allowing the water molecule to move across the membranes. And such a diffusion direction is depends on osmotic pressure. Okay, any questions so far? No. Okay, now we talk about the ATP power pump. Basically, it's need the ATP hydrolysis to generate the energy to deliver the molecules against its electrochemical gradient. So the ATP, this is the chemical structure of the ATP. There will be a T is a three means three phosphate group. Okay, phosphate group chemically is called a PO4 negative. Okay, so it has a highly negative molecule. If you put three highly negative ion together, you need a lot of force. So there are a lot of power in this chemical bond. Once you break down those power, it can generate a lot of energy surrounding here. So that is that's the cell. That's the strategy cell to utilize those energy. Okay, so those energy kind of can change their conformation and help those ATP power pump to transport molecules. Okay, so ATP power pump have a multiple family or multiple class. So it's a P class pump, V class pump, F class proton pump in the ABC superfamily. So for these three, pump only can transport ion, only can transport small guys, ion. But the ABC superfamily, they can transport a lot of different things. They can transport drug, they can even transport molecule, sugar, peptide, and a lot of things, okay? But those three guys only transport ion, okay? So they are the guy who generate those electrochemical gradient. For example, if you check the concentration of a different ion inside the cell and outside the cell, which is blood, okay, you can see quite different between these two pools. Okay, for example, the calcium, which outside cell would be 1.8 millimole, but the inside cell would be really, really low concentration. Okay, and those calcium can kind of, on the textbook, we will read this, releasing the calcium from sarcoplasmic reticulum can induce muscle contraction. Let us just take a look of this video to see how calcium to trigger the muscle contraction.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend, and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Okay. So just quickly remind you. So why we can control muscle, that is because our mind will activate certain neuron circuit, which will release the neurotransmitters and come from the neuron cell connected with our muscle. And those will activate sarcoplasmic to release. There are a lot of calcium stored inside the sarcoplasmic reticulum, which is the spatial organelle in the muscle cell. Okay. Then, if the neurotransmitter will activate will tell those sarcoplasmic release the calcium to the cytosol. Okay, and the calcium will bind to those filaments and bind to troponin, which will change their conformation, then release the binding site. Then the myosin can bind those binding site and start to do the muscle contraction. Okay, and the point is why the cell can store so many calcium ion in the sarcoplasmic reticulum is all driven by those calcium ATPs or ATP power pump. Okay, so this is the member of the sarcoplasmic reticulum, the gray layer, and the up pool will be the lumen, and the lower will be the cytosol. Okay, so how this calcium ATPs can generate such an electrochemical gradient here, or means how can they keep transport a lot of calcium in the lumen and the storage and generate such an electrochemical gradient. So there's an ATP binding site of those ATPs and phosphorylated aspartate here, and there will be two calcium binding sites here. Okay, so first they will have two conformation, the E1 and the E2 conformation. So in the E1 conformation, such a calcium binding site have a higher affinity with the calcium. Okay, and the ATP will bind there, and the ATP hydrolysis will happen, and those aspartate will be phosphorylated. Why this called a P-class pump is because all these member contain conserve aspartate, which can be phosphorylated. And such a phosphorylate will change then from the E1 conformation to the E2 conformation. Okay? Then for the E2 conformation, they will open to the other end, and the calcium, under this conformation, the calcium binding site affinity will be lower. That is why they will release the calcium. Okay? Then those phosphate group conjugate to the aspartate will be released, then they become the E1 again. So by doing this, they will keep changing their conformation through the ATP hydrolysis and the phosphorylation, and change their conformation and deliver the calcium from here to the other side, and generate the strong electrochemical gradient in the SR lumen. Okay. So this is the cation for the calcium ATPase, but this is the real protein structure of those calcium ATPase. So this is the calcium binding site, and this is a phosphorylated site as parted, and this is the ATP binding site. Okay, so they will change from this conformation to the other conformation. Okay, so release the calcium, and calcium can go to the SR lumens. So by doing this, there are a lot of calcium ATPase localized to the different organelle. For example, they localize to the plasma membrane, they localize to the mitochondria and the ER membranes. They will keep pumping calcium to the matrix of the mitochondria, to the lumen of the ER, and to the extracellular space. After a while, the calcium in the cytosol will be really, really low. That is, and the ER lumen and the matrix or extracellular space will be kind of calcium abundant environment. So in the cytosol, because it's really low, that is why a little bit calcium going to the cytosol can serve as a secondary messenger. Okay, the cell has the mechanism to sense such anything or any calcium going to the cell can serve as a trigger to activate certain signaling. Okay, like a muscle contraction. Okay, yeah. And many ATPase, they share the really similar protein structure. For example, this sodium potassium ATPase showing in the gold colors, and the purple colors show the calcium ATPase. They overlap a lot. So they share the really conserved structure. They look really similar, okay? So that is why if you check, this is the sodium potassium ATPase. They look really similar to this, right? Yeah, all of them P-class pump look really similar, but they have inside the protein, they have a different ion binding site. So this is the sodium potassium ATPase. So they can generate the stronger or transport sodium to this side and the potassium to this side. So in this sodium potassium ATPase, they have sodium and the potassium binding site. In the E1 conformation, they have a stronger sodium affinity, lower potassium affinity, okay? And the ATP hydrolysis happens, phosphorylation happens, and change to the E2 conformation. In the E2 conformation, they have a lower calcium affinity, but higher potassium affinity. That is why you will absorb the potassium and release the calcium. And after that, the phosphate group will be removed and come back to the E1 conformation. Then it will kind of release the potassium and again to collect the sodium. So keep doing this a cycle, it will accumulate more sodium here and accumulate more potassium here, okay? With really similar mechanism. Okay, so those are P-class proton pump. P-class ATP pump. Okay, so next, this is a V-class proton pump. So why it's called a V-class proton pump? Because you can find those proton pump mostly to the vacuum or vesicle or lysosome, some kind of vesicle like a organelle. Okay, so inside those vesicle or vacuum, this is the V-class proton pump. They will use ATP hydrolysis as energy to keep a transport proton into the lumen. Okay, but you may notice that what's the difference between the A and the B? In the A, they will transport a lot of proton, and in the B, it will transport a lot of proton and also transport a lot of chloride. Only in this case, it can make lumen more acidic. But in the case of the A, the pH still keep neutral. But when I first started read this textbook, I think it is really weird, because what's the definition of the pH? The pH definition depends on the level of the proton. The more proton mean lower pH level, right? So why you transport a lot of protons still cannot make a pH level lower. That's weird to me, okay? So now I would like to explain this. So in this case, if you just use this ATP V-class proton pump to transport a lot of proton from this side to the other side, it will generate a lot of electrochemical gradient of the proton, okay? And there are a lot of proton unipolar or proton ion channel, whatever, they will, once you generate such an electrochemical gradient, they will really quickly leak out. And it cannot keep enough amount of the proton in the lumen. And such an ATP power pump, they deliver the molecule really slowly. It just delivers kind of usually 1,000 molecule per second, but for the diffusion, it can achieve 10,000 per second. Okay, there's no way they can accumulate enough amount of the proton to achieve lower pH, to make this lumen environment more acidic. So the only way is you need to remove such an electrochemical gradient. So in this case, if there's an ion channel, chloride ion channel deliver the chloride, the proton can combine with the chloride and form the HCl. That will kind of take out the charge, and there will be no more electrochemical gradient. And then there's no driving force to make a proton leak out. Then they can make this lumen more acidic. Okay, you need to deliver the proton and the chloride together. Otherwise, the proton will leak out. Okay, yeah. Okay, so this will be the P class, V class, and the F class proton. Then we will talk about the F class proton pump. Okay, once we finish this part, we kind of may dismiss today. Okay, so F class proton pump they have the other name is ATP synthase, which is the enzyme cell mainly used to generate the ATP. Okay, so our energy comes from the food we eat. Okay, so the food we eat, it will go through the glycolysis. So the sugar, the chemical bond between the carbon of the sugar has high energy. So breakdown of the bond can release the energy. So they kind of release the high energy electron. So those electrons will be captured by several chemicals called NADH or FADH2. Those are chemicals will receive those high energy electron during the glycolysis. Okay, so NADH and the FADH2 then will go to the mitochondria and release those high energy electrons to change a lot of different proteins conformation, and those we call the electron transport chain. They will change their conformation and keep pumping proton from the matrix to the intermembrane space. Okay, and it generates strong electrochemical gradient of the proton here. Okay, then those F class proton pump can use this proton electrochemical gradient to kind of to change their conformation and generate combined ADP with the organic phosphate group to form the ATP. Okay, they can shift the energy from the glycolysis to the ATP. Then the cell can use this ATP to do a lot of different work. Okay, so why is called so this ATP synthase is F-class proton pump. And just keep searching why it's called F-class proton pump. It's because it's an energy coupling factor in mitochondria. I don't know why it choose F as a F-class proton. Doesn't make any sense, right? Yeah, it could be E-class, C-class. Why it's called the F-class factor in mitochondria? It's so weird. Okay, no matter what, it's an F-class proton pump, but they are ATP sensitive. If you say I'm doing the F-class proton pump, probably no one can understand what you are studying. But if you mention, I study ATP synthesis, oh, that's ATP synthesis. Okay, yeah. Why it's called the F-class proton pump? I don't know. It's because of factor, okay. Okay, so let me just take a look of this, how the proton electrochemical gradient can drive this ATP synthesis to generate ATP.
ATP synthase is a molecular machine. The that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized. Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft.
Okay, we stop here. So those are chemical cartoon of the ATP synthesis. So there's a proton binding site will allow the generate a tunnel for the proton flow. It will go, and those proton flow will rotate this the whole gamma subunit, and those gamma subunits will change the conformation of those alpha and beta, those kind of bow, like a pattern, like a protein. So from the top view, this is alpha, beta, alpha, beta, alpha, beta, okay? And this is gamma, okay? So they rotate and it will change. The alpha, beta, they work together, okay? So there was three pair change their conformation, and another ATP will bind here. So they will change among the three different conformation, O conformation, L, and the T conformation. So for the O conformation, it stands for the open conformation. So in this case, they have a binding site allowing the ADP and the phosphate group can freely go in, and the ATP can go out. Okay, here. But in the loose conformation, ADP and the inorganic phosphate were loosely bound there. Okay, here. Okay, and for the tight conformation, they can force the ADP and the inorganic phosphate get closer. And the people believe this can spontaneously force them to form the ATP, okay? Yeah. So after that, it will change to the O conformation again and release the ATP. So by rotating this gamma subunit, those alpha beta pair, they will change continuously through the OLT, OLT, they can generate the ATP, okay? So so one turn will generate 3 ATP, okay? Yeah. Basically. So, and also this is a smart machine actually. If you generate too much ATP, they will stop rotating. They will stop use proton as to rotate their these gamma subunits. They will reverse the orientation and stop pumping. They can also pump in the proton back to this site. Okay, if they think I have too much ATP, I can pump proton back to the intermembrane space. Okay, yeah, but if I don't have enough ATP, I can use a proton follow its electrochemical gradient to generate the ATP. So if you add ATP to this subunit, you can rotate this structure. So people, so at that time, in the early days, this is a hypothesis, right? People want to know is it really, they can really rotate. So many Japanese researchers, they just purify the alpha and the beta and the gamma subunit and the coating them on the cover glass, and they try to add ATP to see whether they can rotate. But if this gamma rotate, you probably cannot tell because it's only one spot, right? So they just conjugate a lot of actin filament on this gamma subunit. So you can see once they add ATP, this actin filament just start to rotate like a helicopter. Yeah. Okay. Yeah. To prove this. Okay. So those ATP synthase, you can find mainly. Actually, you can find to the many side, but mainly localized to the mitochondria and the chloroplast in the platelet cell. Okay. So they will either localized to the inner membrane and will use the electrochemical gradient from the inner membrane space to generate to the matrix to generate ATP, or in the chloroplast, those are ATP synthase will localize to the thylakoid membrane, and they will use electrochemical gradient will be higher. The proton will be higher in the thylakoid space and compared to the stroma. So you use this to generate more ATP. Okay, so this will be the F-class proton pump. Since we only take a 5-minute break before, so we will dismiss earlier today. So next week we will introduce the following section and also introduce the chapter 20. Any questions so far?